Alright, so what are some 5 main causes of karyotype abnormalities? Remember, karyotype is just our chromosomes, so we can get this thing called polyploidy or aneuploidy, and we'll go into that more. So the first thing to do is you need to understand prophase, metaphase, anaphase, telophase. If you don't, the Khan Academy video should help clarify those different phases. Now that you understand what the different phases are, let's talk about anaphase lag. So what can end up happening in anaphase lag is that as the microtubules are attaching to the centromere, essentially sometimes they may not attach properly, or they may not pull the chromosome to the opposite poles. And as such, one chromosome will fail to migrate. I've drawn this one as the one that's failed to migrate. And as such, it ends up creating this micronucleus. Now, this is an abnormality, so the body decides to degrade it. But what ends up happening is that, well, this division has the proper chromosomes versus this one actually lost one, right? So that's that. And essentially what that means is that your anaphase lag could actually lead to monosomes, so only having one chromosome from the male or female, from the opposite sex cell, essentially. Because what ends up happening is you get a blank like this, and so the sperm comes along, for example, and only it is providing the chromosomes. In that example of abnormal cell division. Therefore, abnormal cell division can only lead to monosomes. It couldn't lead to trisomy because it has nothing to do with one chromosome joining the other. It has to do with them splitting and one just not going to other cell division and being degraded on the spot. So now we're going to talk about non-disjunction. And non-disjunction is basically we have failure of our sister chromatids or chromosomes to separate. And there's going to be two types of non-disjunction. In meiosis 1, remember we have the two chromosomes and we're splitting the two chromosomes themselves, right? So what ends up happening is that if they don't split, let's say one goes with this one, so we get essentially trisomy here, versus here we're going to get a monosomy because only the sperm in this example, assuming this is the sex chromosomes, can provide the DNA. So 50% of our result is going to have trisomy, these two right here, because they have two strands here versus we only want one sister chromatid, but we have two here, plus the sperm, so we end up getting three. We have nothing here, so the sperm comes along, we get one sister chromatid, and then same thing here, the sperm comes along, we get one sister chromatid. So in meiosis 1, non-disjunction, 50% trisomy, 50% monosomy. Now in meiosis 2, we've kind of already split our chromosomes, and now we're going to split into our sister chromatids right here. But what would end up happening is that for one of them, you would end up having a proper split, right? So you'd have a proper split right here. And then for the other ones, because you're having non-disjunction, so let's say both of these two sister chromatids come along, you would end up getting two of them here. So you'd get 50% normal because let's say this one is split, so one sister chromatid goes here, one sister chromatid goes here, you get a 1 and 1, that's fine, it's normal. Let's say this one has a problem. So both sister chromatids travel to the opposite pole, this pole gets nothing, as shown here, versus this pole would get both of them. So 50% abnormality in meiosis 2 non-disjunction versus they're both abnormal in meiosis 1 non-disjunction. Now, you can also have this thing called meiotic failure, and it is just complete non-disjunction, which is the cells just fail to separate. If that is still complicated, maybe a Google search will help. To me, it makes sense. Now you're gonna have dispermy. So this is essentially just the simultaneous fertilization of the egg. So usually, because we have our fast and slow conduction blocks, this prevents dispermy. However, here, because we're having two sperms fertilize one egg, we're getting triploidy, not trisomy. So trisomy is where one of our chromosomes has an extra chromosome, for example, right? That's going to be trisomy. So let's say our instead of having an XY, you can have an XYX. That would be trisomy. Versus triploidy is that all our chromosomes, 1 to 23, they all have an extra one, right? So it's like a whole new genetic pool is being added. And if you look at the karyotype, you'll see that each chromosome, 1 to 23, has an extra one attached to it. So basically, there's a trisomy on 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, all the way up to 23. Versus trisomy itself is only just on one of them. So now moving on to incorrect DNA repair, you have these 5 major types. Let's start off with the easy ones. You can have deletions and duplications. Super simple. Yeah, and now we're gonna start talking about the more, I guess, complicated things, which are gonna be inversions. So there's 2 types, pericentric, paracentric. Paracentric means same side. Pericentric means different side. I don't have colors, so I've just put shapes here. Essentially, if it's pericentric, this triangle here has moved across the centromere to the other side. So it's gone from the Q arm to the P arm and vice versa. Versus paracentric, essentially the Q arm, let's just say this is the Q arm, has stayed on the same side of the centromere, right? Now for translocations, this is where we're going to be inserting genetic material from one chromosome to the other. And as such, there's going to be 2 types. One's going to be insertional, the second one's going to be reciprocal. So if I have chromosome A, it's sending just its DNA to, or genetic material to chromosome B, so it's losing. There's no net loss, but chromosome A has lost some and sent it to chromosome B, versus reciprocal, maybe chromosome A sent some to chromosome B sent the same amount or a little bit less, a little bit more to chromosome A, so that's the difference between that. Now for Robertsonian translocation, this is quite interesting. Essentially, remember our acrocentric chromosomes, they have those very tiny arms right here. What can end up happening is that because they're so small, they can end up just fusing on top of each other. So this one could fuse with that one, and you end up getting this very long chromosome. However, it's still a normal karyotype because you actually haven't lost or gained any DNA. So in terms of genetic DNA, this is the same thing as this. However, all we've done is we've fused it over, and the lecture goes into more about this. So essentially that's that. We have our additions and deletions. You also have this thing called a ring chromosome. Remember, the telomeres are the tips of our chromosomes. It's a B. And so what can end up happening is that they can stick onto each other and create this ring-like shape. And yes, you can also lose some DNA through this process. So to summarize, anaphase lag becomes monosomy because we have that degeneration of the micronucleus. The chromosome missegregation can lead to monosomy, trisomy, and triploidy. Meiotic failure can only lead to triploidy. Dispermy can only lead to triploidy. Again, 2 sperms, 2 whole new genetic profiles being added to the egg. Therefore, triploidy, you're never going to get trisomy from dispermy. Meiotic failure, essentially this is like a complete non-disjunction. So this is where everything has been pulled over into one pool. Essentially, you're creating a type of dispermy here, hence that's why it's leading to triploidy. In terms of chromosome missegregation, this is very broad. This can encompass non-disjunction in meiosis 1, meiosis 2. Therefore, we can really get monosomy, trisomy, triploidy. So this is a good summary table and probably will come up. Near 100% chance it will come up. So for aneuploidy, essentially this is addition or loss of some chromosomes. This is usually from non-disjunction. Versus polyploidy is a whole new set of chromosomes being added. So polyploidy is really just aneuploidy adding onto 1 to 23 of our karyogram. So essentially polyploidy, it usually happens from complete non-disjunction. Remember, Polyploidy would never happen from anaphase lag because anaphase lag only leads to monosomes. Essentially, your cells are failing to separate, and then dispermy is really the most, I guess, common sense one of leading to polyploidy. Triploidy is a type of polyploidy. You can also have quadruploidy, but that's very rare. You also have microdeletions. They can be diagnosed with FISH. So FISH is this technology which essentially we get the, we get the karyogram, we get the chromosomes, we multiply them out exponentially, and then we use fluorescence to basically figure out a ratio between them. And by doing that, we can kind of figure out where microdeletions have taken place. Now I wanted to talk about the GLS diseases. There's quite a lot of them. I think there's like 10 or 11, something like that. Yes, you need to know all of them. You need to know all of their karyotypes. You need to know all of their, not all of their clinical features, but the key clinical features. Luca's notes do quite a good job on that. So essentially you need to know all of these. So let's just go into like triple X, some key clinical features, hypotonia. This is decreased muscular tone. On exam, you can't say decreased muscular tone. I mean, you can, but it doesn't really show understanding. You need to know the proper words. For example, let's say this is like easy MCQ, easy EMQ. You'd be leaving marks on the table by not doing this, and it's in week 2 as well. So you have like easily 10, 12 weeks to go over this. Maybe a karyotype for diplo Y could be 47XYY. Maybe MCQ, like it could be a picture of this and then what is it? So it'd be diplo Y. And then some key clinical features. Notice how there isn't a lot. There's only 1 or 2 key clinical features. There's a lot of features such as macrocephaly, such as enlarged head, that kind of overlap. But here we have prominent glabella, so protruding frontal bone situated above the root of the nose. The GLS was so good for this week, and it really focused on this, so I do that.